So I'd just like to welcome NT Fisheries today. We have a suite of lovely speakers. We have Laura, Alex, Deepak and Danilo, who will be presenting to us today about marine research and monitoring up here in the NT. So welcome.
Hello everyone. So I'm Danilo. My colleague and I, we're going to explain. We cannot see anything.
You should just be able to step to the next slide.
Yeah. Okay. So we're going to present the research we are doing at NT Fisheries. First of all, I want to acknowledge the country. We respectfully acknowledge First Nation people and recognize their continuing connection to the land, water, and communities. We acknowledge the Larrakia people as the traditional owner of the land, seas, water, where we are meeting here today, and the enduring connection to the ocean, river, and mangroves that sustain our fishing population. We pay our respect to elders past and present and extend that respect to all Aboriginal and Torres Strait Island people here today. Who's who in NT Fisheries? So we are a group of researchers that have different skills, and we try to collaborate with each other to make the best output of our research. We are led by Michael Asher. I'm not going through all the names here. I just want to mention that are missing two people. One is Joe that moved the department temporarily, and the other one is Amy that is in maternity leave. So who am I? I did my studies in Italy, my country, moved to Australia to do the PhD at James Cook University, moved back to Italy to do my postdoc, and now in 2024 I got the position here at NT Fisheries. I have experience in different fields, from trophic ecology, biomass reconstruction, modern catches and behavior, movement ecology, marine litter pollution, visual sensors, some DNA research, environmental impact research, etc. What I'm doing today at NT Fisheries. So my recent position, I'm looking after the coastal line fishery. Coastal line fishery is basically a fishery fishing in the coastal area, mainly using hook and lines. Is divided in three different sectors: commercial, tour operator, and recreational fishing. And the main target are blackgill fish and gold snapper. Secondary species are grass emperor and stripper snapper, and then there are a bunch of other tertiary species. Part of our job is to monitor the fishery and to collect some biological data. This data then used for stock assessment. Based on the sector, we collect different information. For example, in the commercial, we collect frames and biological samples. In the FTO, we have an observer program, but also, when possible, we collect frames. In the recreational sector, at the moment, we are doing just recreational surveys, but also in the near future, we are implementing the frame collection and angle diaries as well. Recreational fishing is also part of my job. Why the recreational fishing surveys are important? Because they quantify the recreational fishing pressure. They are used for informed stock assessment and management, and also support the sustainable fishing management. What data we collect? Mainly catch and effort, species composition, and when possible, we collect also biological data. To do the recreational fishing surveys, we use the Stratify survey design to estimate catch and effort. We conduct this one mainly in the boat ramps. Based on the effort, we divide the boat ramps in two parts, primary and secondary. In the primary, we conduct the actual survey. In the secondary, we counted the trailers and estimated the catch and effort from the number of trailers. Other part of my job is also monitoring the reef protection area. NT has 5 main reef protection area. This area had been designed to protect gold snapper and black fish, allow them to reproduce and move in other reefs as well. So our job is to monitor the abundance of the fish inside, outside the reef protection area. We mainly monitor the 3 reef protection area close to Darwin. So Lorna Shoal, Charles Point, so here. Lorna Shoal, Charles Point, and Basses Island. We also implemented our monitoring system using stereo browse. Stereo browse basically using 2 cameras and a special software to calibrate the cameras and get the best precise measurements. Other part of my job, so I'm part of the gold snapper recovery plan team. In the last assessment, the gold snapper was declared depleted, and because it's minimum below the minimum stock size threshold. So we don't know many things about the gold snapper, especially in biology, ecological space. And part of my job is to do some research on gold snapper. So this is, we secured 2.5 million over the next 2 years for gold snapper research. Some area that we need to improve are maturity, movement ecology, post-release survival, genomics, diet, and citizen science. So there will be potential for some research in this space. So if someone is interested, just drop an email or get in contact with us. Thank you for your attention. There is a QR code for my publication list. And I move to Alex. Thank you.
Thanks, Danilo. So hi everyone. My name's Alex. I'm the fisheries enhancement scientist for NT Fisheries. And following on from what Danilo was going through, I'm going to run through kind of the research and monitoring initiatives from NT Fisheries in that space. So fisheries enhancement, NT Fisheries has been doing a lot of fisheries enhancement for quite a few decades. The main reason for that is to support recreational fishing and the territory lifestyle. The 3 main components for NT Fisheries fisheries enhancement packages: artificial reefs, FADs, which are fish aggregating devices, and also fish stocking. We've been doing it for a long time, but in the past couple of years, especially spurred on by the golden snapper, there's a much larger focus on the research and monitoring component of fisheries enhancement, especially coming up in the next few years, which is really exciting. So I'll start with artificial reefs. So for people that aren't familiar with artificial reefs, they are a reef, but they're made by people. They're ideally designed to produce their own fish and act as functional habitat. NT Fisheries deployed 4 artificial reefs in 2019. They use these reef pyramids that you can see here, which are massive concrete structures. These are about 5 meters high. They deployed them in around Darwin and off Dundee, and they get a development. They develop a benthic invertebrate community, and then the idea is that fish will then colonize them and start to use them like they would a natural reef. NT Fisheries has historically monitored these reefs with brubs, which are beta remote underwater videos. It's basically just a couple of GoPros with some bait in front of it. But a lot of artificial reef monitoring is heading towards ROVs, so remotely operated underwater vehicles. And this is mainly to capture the cryptic species. So you can see here, this is the reef off North Gutter. After 1 year of deployment, you've got some snapper there that are kind of cruising around. And after 2 years, when you've got a lot of colonization, you do have some of those more cryptic species like coral trout that live inside of the artificial reef. When you use brubs and you just have the bait sitting somewhere around the artificial reef, you don't necessarily capture those species, whereas with an underwater vehicle, you can actually go and look for them and find them. These artificial reefs are fished, but the next phase of the NT Artificial Reef Program is to deploy an artificial reef in a protection zone. So hopefully that'll happen towards the end of this year. And that again ties into the golden snapper to support the reef fish recovery efforts that we're making. The next phase, the next thing that I look after is FADs, so fish aggregating devices. These are basically just buoys that are sitting about 15-ish Ks, 15-ish nautical miles, sorry, off Darwin, and they're designed to attract and aggregate pelagic fish, which are more sustainable and kind of offset pressure off our more vulnerable reef fish stocks. We deployed 4 FADs in December last year. Fisheries has deployed FADs many times in the past, and the consistent theme was they all got blown away, and none of them lasted for more than, I think the longest we ever got one was 1 year, but most of them only lasted a few weeks to months. So after consulting with other jurisdictions like WA and Queensland, who have big tides like we do, where the solution was to just throw all chain underneath it, no rope, just chain. And the good thing is we deployed them before the wet season and they're still there. So they're getting pinged constantly by with a GPS tracker. So we always know where the FADs are. Monitoring them, we mainly use the boat ramp surveys that Danilo was talking about and try to use kind of harness the power of recreational fishers and use citizen science. Monitoring FADs on your own using video surveys is pretty difficult because they don't always hold fish. Fish come and go, and you don't want to drive all the way out there, send a bunch of people to go out and go, oh, there's nothing on the FADs today. Well, get back to the office, I guess. So we want to go use fishermen, which is something that we've heard from the public, is they want to be involved and help us do our research. Oh, now, sorry, and also go, oh, there is sound. So this is a video from March, which it's not a great video, it's quite small, but we did, when we watched them a few months ago, sorry, we did find a school of rainbow runner, which are actually quite a rare fish to find up in Darwin. They're about this big. So really exciting coming into the build-up where we get more calm weather. We're hoping that a lot of people will start catching fish off the FADs. I know that people have been catching Spanish mackerel off them already, and I'm hoping to get one this weekend, so I'm going to the FADs this weekend. The last section is fish stocking. That probably takes up most of my time. NT Fisheries has been stocking fish around Darwin since 2004. Most of that is through barramundi stocking, and that is to improve fishing opportunities and enhance that territory lifestyle. But we're now planning to start or restart a golden snapper stocking to support the wider recovery plan for the species. All the fish are grown at the Darwin Aquaculture Centre, which is where my background is. Did a PhD in aquaculture, and I've been started there as a senior technician, then was as an aquaculture scientist. So it's a great facility, and we're able to produce a lot of fish. Barramundi stocking, most of the barramundi that we stock go into Manton Dam, but we also stock dams and lakes around the Greater Darwin region. A big one is the Palmerston Lakes, so people who live in Palmerston would know all about that. The stocking program facilitates the competition that gets run every year, and we get great engagement. We see kids running around all the time catching fish, and we also get recaptures up to about 60%, which is fantastic. We monitor our wider stocking program through tagging with AFAN, so they have their own tagging program, which we kind of feed into. Fish get green tags, as you can see in the top, and then hopefully grow into big ones. Like you can see down at the bottom, you can see it. That's a big barra out of Manton Dam. If you do happen to catch a barra with a green tag in it, please report it to AFAN, because I do check the database to see if anyone's been catching them. So finally, golden snapper stocking. So like I said before, this is a new project to complement the wider recovery plan and the other enhancement programs. People love fish stocking, so there's a lot of social benefit as well. It's a new species for DAC. We did do a bit of it in the 90s and a bit of it in 2017, but we really were starting from scratch from an aquaculture perspective when we did our first season last wet season. We learnt a lot, and we're able to increase the number of fish produced by an order of 10 each time. And we've got our next run in 3 weeks. So hopefully we're going to produce a lot of fish. The plans are to really ramp up production and try to stock these fish around Darwin Harbour. The monitoring program is going to be a big part of this, and that's what I was talking about, the future, how we're really putting a focus on it in the future. We're going to monitor these guys mainly through genetics, so using DNA and genomics and SNPs to identify which fish that are getting caught in the wild are from hatchery origin or not. We might also use different tags to track larger fish. So we can tag larger fish, release them, and then again use that citizen science aspect to see if we can get recaptures there. I'm going to hand over to Laura, who also knows a lot more about genetics than I do. She'll also cover some of the more in-depth details on SNPs and golden snapper.
Morning, everyone. I'm Laura. So a little bit about me. I thought I'd include some background. I'm relatively new to NT Fisheries. I started last November, coming from the University of Queensland, where I did my PhD on sailfish, and that was largely in population genetics, but I dabbled in a few other things as well. Very passionate about citizen science research, and I did a bit of ecological modelling, which it helps now that I'm looking at fisheries modelling. And prior to that, I had some policy roles with the Queensland government and was part of their government graduate program. That was 10 years ago now. Now here at Fisheries, I am continuing to use sort of my genetics and trying to get some projects running using genetics, including golden snapper, and then looking after the barramundi and Spanish mackerel fisheries. So as Alex mentioned, and I thought I'd talk about it first, we need to monitor the stock golden snapper once we get to the point where we can release them, because Alex and his crew have been spending a lot of time trying to grow them and keep them alive. But, are they still alive and contributing to that population once we release them into Darwin Harbour is really the big next question. And so for that, the key thing is, can we identify if we recapture a fish, is it from a hatchery or is it just another wild golden snapper? And thankfully, if we have the genetic profiles of the broodstock parents, we can use relatedness, the genetic relatedness and their kinship coefficients, which I've got in this lovely pedigree chart, to tell whether those broodstock parents are the parents of that recaptured fish or whether they're an unrelated wild fish. So that's the main idea there. So at the moment, we're just in the setting up stage, but hopefully once we get sort of that citizen science fish frame program started up, we can start getting those genetic samples and come up with some quick tests to be able to tell whether or not they came from the hatchery. But yeah, that's very long term. And so I thought I'd also just generally talk a little bit about how we monitor fish stocks, in particular using genetics and also odorless, because we do a lot of that with the barra fishery. And then I think Deepak later will kind of go into a little bit more detail on how we use that information for stock assessments and management. So in terms of defining a fish stock, I guess also in terrestrial ecology, we sort of need that spatial management unit to see if we're managing things at an appropriate scale. And so for fish stocks, I guess like other terrestrial populations, we use biological information to try and define that, and ideally multiple lines of evidence. But also there's sort of political and practical considerations that I'll talk about a little bit in the next slide that inform how you define a fish stock. So when it comes to their biology, what are we looking at? What kind of information do we need to get from their biology to define a stock? So whether they live in a particular spatial extent, what is that spatial extent? Whether they're mixing with fish from different areas in particular, that'll help define those boundaries. Whether fish in a particular area have similar life history profiles. And then of course fish spatially, whether they're having, whether they will respond the same way to fishing exploitation. And so in terms of all the different factors that kind of lead into defining a stock, as I mentioned, those multiple lines of evidence, there's a few different techniques we can use. So genetics, parasites, otoliths, the morphometrics, and sort of the knowledge about the life history from different areas. But then also leading into those political considerations, we have like the jurisdictional boundaries, socioeconomic factors. So a lot of the time, sort of state boundaries will dictate how we draw our management of fish stocks, and then sort of practical things about the fishery, data availability, where the fishing grounds are, and those sorts of things as well. So how can we use genetics to try and define stock boundaries? So nowadays we are using, I think, as Danilo mentioned, single nucleotide polymorphism SNPs. So that's looking at the whole genome. Previously, we would just look at a little section of DNA. If you guys are familiar with genetics, and compare like maybe 600 base pairs, whereas now we can use thousands of little differences across the genome. And so what that tells us is, I guess, like the evolutionary sort of history of the fish. And when we are looking at those older methods, that might be telling us things on a scale of tens to hundreds to millions of years, whereas when we're looking at SNPs, we can kind of tell a little bit more about the recent evolutionary history there, so more on the scale of like tens of thousands of years. And so that's really why it's important as well to use other techniques than just genetics to define fish stocks, because some of those other techniques are going to tell you more contemporary things about what those fish are doing. So if we're using like tagging and movement studies, like otolith chemistry profiles, that tells you within the lifespan of the fish, whereas this is across quite a few generations. And what's really good as well about those high resolution markers is that we can pick up quite small genetic differences. So fish tend to have quite a lot fewer barriers than terrestrial animals, and so they can mix a lot more easily. This is a generalization, not all fish do this, but I looked at the sailfish in my PhD, and that's the example I presented here on the slide, and they are highly migratory. And so we are looking at a scale of, as you can see here, like ocean basins and globally across the tropics. And so, but we can still pick apart some of those differences across those populations. But what my study did actually show, which was quite surprising, is that we have quite a lot of connectivity across the Indo-West Pacific. And then those implications then for management is that those regional fisheries management organizations, which you can see they've got like, I guess, ocean basin jurisdictional boundaries, they need to work together to manage the fish that are moving across those areas. So otoliths, for those of you who don't know, there's an ear bone inside the fish. I think many of you people do know. So we use them for aging. They, in most species, have annual growth increments. How defined they are sort of depends a little bit on the species and where they come from. But this gives us really crucial age data that we can use in stock assessments, and size at age in particular. We use them to infer growth and calculate growth curves. We can also look at microchemistry, so changes in chemistry. The chemical profiles over time can tell us things about their life history, and can also tell us if there's spatial changes as well. And the chemistry profiles can tell us about their stock structure. And otolith shape as well is another way of looking at that. So yeah, I've got some growth curves there. And then this is from Brian's paper looking at strontium ratios that sort of show the difference between fish that remain in fresh water and fish that is moving between fresh and salt. And with that, I will move on to Deepak. Thank you.
Thanks very much. Good morning, everyone. So, I work as a senior research scientist with NT Fisheries. Just want to start with showing where I come from, and now I'm in Darwin. So I originally come from India in southern part, where I did my Bachelor of Fisheries Science. Probably surprising, you'll see that it's a university dedicated for fisheries science, and there are multiple universities. There are around 1,000 graduates. Pass out in a year. So there's a lot of graduates just focusing on fisheries science. I specialized in fisheries resource management for my master's in Mumbai. And then I moved on to Ireland to do my PhD again in fisheries. And that was more to do with fisheries stock assessments. And then I landed in Darwin, worked for Pasquale in pearl oyster aquaculture for 6 years. And then in India fisheries for the last 5 years. Back to where's my expertise, essentially. By the way, yeah, my last name is a tongue twister. If you're brave enough, you can try. So we've been hearing a lot of research from the coastal area. So I do a lot of research in the offshore region. So and particularly in the offshore snapper fisheries, which is essentially combined Timor Reef fisheries and domestic fisheries. The Timor Reef fisheries, the fishing happens in the purple box there, which is called a Timor box, and it's primarily use mobile fish traps to catch fish. And the rest of the area is the fishing allowed for demersal fisheries, and they primarily use the bottom trawls. The four major regions here, as most of you probably know, Joseph Bonaparte Gulf on the west side. Then you have Timor Seas, and then Arafura Seas, and then the Gulf of Carpentaria. The three target species in these fisheries: the goldvine snapper on the top, saddletail in the middle, and crimson snapper at the bottom. In addition to this, there are a lot of bycatch species that comes to the trawls. The objectives of ND Fisheries as a regulatory agency is to ensure the sustainability of fish stocks. So we do a lot of fish stock assessments, our core research work, but also we try to define what are fish stocks because they can change over time. So stock structure studies also becomes something that we constantly carefully look on how things change over time. I just want to, with this slide, which I just want to show how stock structure studies, the outcomes, how they look like. This is a study I did in 2004 for my master's. So that is a peculiar type of fish. It's called Bombay duck. Obviously, it's not a duck. So, and I had no funding for this research, so we used low-cost methods, so looking at the shape variations of the body. In particular, this is called a trust network, looking at different, so you can see there are 3 or 5 different boxes and diagonals going across the boxes. The fish is not, the distribution of the species also is very interesting. There's a west coast and east coast, but they are geographically isolated. You won't see them below the 15-degree latitude. So it's geographically isolated. So definitely there are 2 different stocks, but my interest was to see whether there are any difference in stock within the coast, within the west coast, if there are more than 1 stock or not. And those are the results. So I know it's not very clear, but yeah, we did see some separation between east and west coast. And there's no clear differences. And that's what we think is this fish have 98% of water content. So it's like very flabby. So it's probably not have a strong, there's no strong features to give that a signal. But we didn't see any difference within a coast, within the west or east coast. So they are, probably had. So what we think is they should be managed separately, east and west coast, but we don't need any additional managements because they are all 1 stock there. So I'll take that from there and show you. We run a few stock structure projects as a collaborator. So first one is the basement jack project, which is looking on the stock structure of mangrove jack. It's led by AIMS. In that project, we use 3 different techniques to essentially have multiple lines of evidence. So we have otolith microchemistry, and then we have fish parasites as a biological tag. And then we are also using genetic information. So this project is still running, so we don't have any results to show at the moment. The other one we just recently established is a crimson snapper project, again funded by FRDC, but also funded by AIMS and CDU. Use the pretty much same techniques, but also a social science component into it. And then we have a small little project running in-house we have brown striped snapper, which we use a completely different technique looking at the shape of the otoliths. So essentially from all these techniques, what we are looking is the phenotypic differences in these stocks, which probably not visible by eye, but there are some phenotypic characters that can be linked to which these techniques can show that differences. So essentially, if we are talking about genetics, there will be some genes that link to phenotypic characters. Why we do stock structure? We take that outcome to do assessments. So the first graph there, the map shows you. So this is a recently released assessment of gray mackerel. And it was led by one of our gold star stock assessment scientists, Sandra. She's not here. So you can see the spatial extent. So that's the area we are looking at. And that is defined by, because we know that's a different stock from the Gulf of Carpentaria. So we use the catch from logbooks from that area. Then you can see the data used are like large amount of data. The second graph shows you this data from 1985 until 2025. And yeah, obviously catches are from the logbook, but there are CPUE, which we use as a indicator for the abundance of the biomass. We also use length measurements and also aging measurements. So, which is, for a long time data sets, you need multiple staff working through multiple generations. So it's an ongoing program. Without that, we can't do any of these assessments. The last slide shows the outcome where the biomass is sitting at the moment. So there's the biomass, in 1985, we think we start assessments at the unfished conditions. So they are like 100% or close to 100% on unfished conditions. And in 2025, around 50%, I guess. So we have a managing target line. So if that goes below that line, we, as an agency, we have to do additional management measures. We also have a project on stock assessment. Stock assessment is not an easy science, it's highly complex. The graph looks simple, but a lot of work go behind it. It need a blend of multiple skills. Then you need expertise in biology, maths, and statistics. And there's no specialist training or university course available across Australia. So a survey in 2023 indicated there are less than 100 stock assessment scientists in Australia. 60% of them are beginner level, and 30% are probably intermediate, and 20% are experts. Now, interesting is that 20% experts are going to retire soon. So we don't have any capacity in Australia to do future assessments. And the industry always asks for giving, we should provide the robust assessments of higher standards, but there's no mechanism here to bring that next generation of scientists. And each jurisdiction do their own things. We don't have communication between us. They do different styles of assessments. For example, NT Fisheries do something, Queensland do something else, but there's no communication between us. So in last year, we established an FRDC-funded project for creating a community practice for stock assessment. So to seek solidarity, help each other, share resources, bring up the science, do more innovations, and then also identify and address common issues. So this is up and running at the moment. There are lunch and learn sessions, which people can join anywhere from Australia. And there are annual workshops happening. So it gives the opportunity for everyone to come together and discuss common issues. You can also be part of the community practice. Two membership styles. So there's a formal membership for people who really want to get into the quantitative, do assessments. But also there's a broader network membership, which if you're just interested to get informed, you can just be part of it. There's no fee for it. They're all free. If you scan that QR code, you can get to the LinkedIn page, which is a good starting point if you're interested. There's also a space called data poor. The assessments which I showed you for gray mackerel, need a large amount of data, but 80% of the stock across the world are data poor. What we do with those stock, because we as, again, agencies have to demonstrate, are they sustainable or not? And it's a challenge when you don't have the data for that. In Northern Territory, we use stock synthesis as a modeling platform for doing integrated assessments or data-rich assessments. So the example of gray mackerel is a data-rich assessment because you have data from multiple sources. You have catch, CPUE, length, and age. In many cases, we only have catch and CPUE because they come through logbook, and having length and age need additional investment. We have to send some staff to the field to collect these measurements, and it's probably not possible to do that for all the stocks or all the species. In that case, we run stock reduction analysis, which we can call data limited. I put a question mark because it's kind of there's no clear definition about what's data limited or data poor. It depends on the country as well. So, for example, when we have only catch data, then we use catch MSY, but also it's not a the outcomes of catch MSY are debatable, sometimes more scrutinized because having of less confidence. But also I want to flag that my expertise is in data poor fisheries because I did my thesis, my PhD thesis. The research question I was asking, where do you start actually if there's no information about a stock, that really you don't know anything about the stock, you don't have the growth information, you don't have any time series, where do you start? So essentially, I'm not going into detail of this topic, but essentially I was looking at simple empirical indicators that you can work out from raw data, such as mean length of the population. And if you start collecting that into the future, how you can start using that trends to monitor the stock, how you can use that to assess the stock, and how you can manage that stock. So that different chapters and that tools are available for that. Future work in offshore snapper fisheries, there potentially you can use artificial intelligence for multiple things. One pilot study with the CDU students, probably in the data science division, we had a few students running AI to automate the collection of fish measurements. We spend a lot of money on staff to collect these measurements, but we are looking at more efficiencies in the space. If we can set up some camera on the fishing boards or other mechanisms, can we make this more faster, collecting more data but also more accuracy? That's other thing. The second application is possibly quantifying the bycatch or discard species in trawl fisheries. There are around 70 to 80 species that usually we see as discards, not in high quantities, but they do comprise a lot of species. But it's probably not practical to collect them through logbooks because that takes too much time. So we need more efficient methods to capture that information. There's also an interest in using AI to detect protected species, endangered and protected species, such as sawfish or turtles, when they interact with the fishing gear. The other area is using, we have started collecting the temperature using some sensors attached to the trawl gear. So this project is called Fish Soup, and it is managed by University of New South Wales, and they have a collaboration with IMOS. You can see the blue sensor is called a Moana sensor. It's attached to all our trawl vessels. Fish Soup stands for fishing vessels as a ship of opportunities. So instead of going on dedicated research vessels, and it's much cost effective if you attach that to a fishing trawl that goes to these grounds every 2 weeks. So it's a more efficient way of collecting the data we need to do further research. So what is different to sea surface temperature is that this is not sea surface temperature, right? This is because it's going with the trawl gear, it's collecting measurements from the same spot they are catching fish. So they're co-collected data. We have catch from our logbook, we have the temperatures profile available with IMOs, and they are publicly available to download. But what we are looking is to how we can use both of this information, catch, effort, and temperature, and what we can do in that space. Probably we can do all of these different things there: habitat suitability, species distribution, how they shift with temperature or climate. We can potentially use them for standardizing the CPUE, also looking at the species assemblage, doing community analysis, and also probably develop some indicators to show what's the state of the fisheries or the whole as an ecosystem. The bottom graph there, you can see is the data collected from 2023 to 2025. So there's 2 years of data. So prior to 2023, there was no data available in this whole region. So there's new data sets coming through, just sitting there for someone to explore. So that these are some collaboration opportunities. NT government also run early career programs. We get graduates every year. Mostly it's through the graduate development program. From the experience, I know most of the people enrolled for this program are from accounting or IT background. There's probably very less from a science or like biology discipline. So if any of your students here, or if you know someone, if you recommend, they probably have a good chance to get into this program. And records show that, previous programs have, 100% of them got employed or absorbed into the system. So there's probably high confidence on getting a job, and also gives the opportunity to see how we work in our agency, identify what skills are needed to get a job in government. That's all. There's a contact there. If you have any need any information, you can contact the workforce. We collaborate a lot of agencies. Some of them are listed here, but also I just want to point out we do collaborate with CDU, but there's potentially we can do more. We have links with AIMS, Australian Institute of Marine Science, and also CSIRO and Museum. Probably we have to do more with Museum. Thanks for listening. I just want to thank all the hardworking technicians in ND Fisheries that collect the data, research data for us. And yeah, happy to answer any questions. I just invite the other speakers here to answer your questions. Thank you.
Yeah.
I just really simple about the fish aggregate, right? So what attracts fish there? Is it just the shape?
So the fish, the way basically, when you put any structure out in the middle of the ocean, fish just try to find any organs. So the difference between our FADs and other FADs though, so on the there's heaps of FADs on the east and west coasts. They're mainly targeted to attract dolphin fish, and in the southern areas, kingfish. Those species are actually attracted to the void itself, whereas up here, while long-tailed tuna, mackerel, kingfish, and Deathledger Valley are attracted to structure, a big part of how they work is attracting bait. So any bait fish will try to find structure that you get little things growing on the voids for them to eat, or it's just something for them to hide against. Anytime aggregation of bait, predators are going to come by. So that's the way they.
Yeah, so thanks very much for your talk. It's really interesting to learn about the diverse science that you're doing. Two-part question. What do you see as the overarching priorities for the next 5 years from a science perspective for those fisheries that you're managing? And what do you see as the social science related priorities? I'm a social scientist. I haven't actually met you in person at Deepak, but I'm one of the social scientists working on the Princess Project.
Yeah.
What sort of you mentioned citizen science, socioeconomic and governance factors are important in strong assessments and hard strategies, obviously. So what do you see as some of the priorities?
To answer your first question, that's probably for Danilo or Alex too, because it's more around the golden snapper at the moment because they are identified as the depleted stock. unless another stock comes in that category, probably your highest priority is how are we going to manage with golden snapper. Yeah, if you want to speak more about it.
So golden snapper projects, I say we have at the moment some planning for the next 2 years, not 5. And we need to improve the research behind what is unknown behind the golden snapper. We are implementing now the research on citizen science with recreational fishes. So probably soonish, we need to start to collect from recreational fishes. Yeah, so we are writing up the plan for this part of the project. Yeah, I don't know if Alex and next 12.
Yeah, I guess where it's exciting, especially for the enhancement project. So a lot of that is to get around help with sample collection. So obviously for DNA, especially you need a lot of fish in one sample, so that's quite intense. If you're doing something with very intensive sampling effort, but if you ask people to donate frames or give them a little DNA collection kits, things like that, it's not only do you get more data, but you also start working with the community and they feel like they're a part of the grand plan and they're included. So it also helps with them like they're being involved, stewardship.
Build trust.
Yeah, builds trust, all that kind of stuff. They see us publishing the results and they know that they were a part of collecting the data for those results. I want clients to be like, yes.
Yeah,
Right.
the component of social science is more important in that space as well, because golden snapper, 95% of the house is by recreational sector. So you need a lot of engagement with people. You can because recreational fisheries are non-monitored in the same way as commercial, where you have logbooks and licensing. Recreational, there's no non-licensing system at the moment, and so it also makes us difficult to track things, how things happen. So I think we probably need more engagement with that public space, yeah.
Just related to that, thanks for all of your presentations. It's always good to hear about everything happening and what you're interested in. But with the golden snapper, I was just a couple of questions. One is, what sort of spatial area are you focusing on for the assessment and for the research? And two, is there any other work happening elsewhere in Northern Australia that can help, I guess, inform the assessment and the status of the stock here, or are you kind of flying on your own trying to understand what's happening?
Yeah, okay. The first question, we were, so the spatial area we look is the Greater Darwin region. Previous research show there are multiple stocks across this region. It's like every 10 kilometers a different stock. Now we can't do assessment for every 10 kilometers. That's not feasible, and that's where we talk about the practicality. So what we do is more what's the special extent probably probably up to the TV islands and around that. So that capture most of the high fishing effort by the charter and recreation sector. So we have a grid system to capture that area. So we do assessments using the data collected from that spatial area. We don't think the fishing pressure is not similar to in other regions, which we combine everything as a regional area of Northern Territory, and so that's separated from the Greater Darwin region. And all the recreational surveys that boat run surveys are focusing on the Greater Darwin region. The second one, I think under 2023 we were kind of doing our own thing. There's some sort of evolution happening in our stock assessments. Stock assessments in Northern Territory started probably in 1990s. They were very simple models, but we, from my understanding, we have invited experts from US few times into Northern Territory to train and build our skills here. So under 2022, we've been standard assessment packages with a stock reduction analysis. From 2023, we started working with other jurisdictions, and we jointly identified this issue with assessment. We don't have any, anyone to depend on or even ask for advice, and that's the reason we created this community of practice. And with annual workshops happening from 2023, we slowly build up our skills. So stock synthesis, which we use for assessing golden snapper, is an outcome from that project, because they did a few training programs to do highly complex models, and now we are capable to do those assessments. And we know people across Australia, and we know who have the expertise if we need to reach out. Yeah.
I have a couple of questions.
Yeah.
So, Alex, bags and artificial reef because they can act as bags. Given that there's potential, I mean, obviously you'll work on a relatively small scale in terms of number of units, but given there's potential for those to enhance overfishing when it comes to training the fish, what kind of management strategies do you have or plan to put in place for that?
So you're right, all artificial reefs act as flats. So basically that's like a common theme. As soon as you put down artificial reefs around the country, first thing you see is pelagians especially just go wood and attach themselves to the same bank. The science around the productivity of artificial reefs is still a little bit grey. It's definitely evidence that they can be productive and designed properly, which is why now pretty much all artificial reefs are going to those purpose-built. They've got lots of different voids. It's not just dumping a boat down or sinking a ship anymore, although people still do that. But in terms of how to stop it just being a fishing hotspot, so the new artificial reef that's going in is going to be a reef fish protection area, and we'll be using our new compliance to try to manage and stop people from fishing it. We're sort of not going to tell people where it is. We'll be saying it's in a big box somewhere. We're not going to tell you where.
Yeah.
But yeah, there's like they can do exactly that. Well, there's no plans to change anything around the existing artificial reefs. Those were put down to enhance give recreational fishing opportunity. But yeah, management and then so was there a second
the second one was thank you, thanks very much. In fact, I was really interested in your old research from India with the Bombay delicious breakfast fish from they go to Bombay. And that they don't go below 11 degrees, even though they probably don't have their own fires. But, and the stocks haven't mixed. I was just wondering about, that was many years ago now, that you were doing that work, and I wondered whether that would change with warming oceans and maybe that come further down as double max, and what phenomena of that kind you may be seeing and how it was doing.
Yeah, these are things which I asked when I was doing the research, but from other research, it's pretty evident now. They are more related to the wide continental shelf in the northern sides. Towards southern side, these continental shelf becomes very narrow. So they don't go to larger depths or anything, and also that low depth kind of give them more sort of food, prey species available more in these regions. Partly also because a lot of river, flows into the northern side or the southern side, so there's it's just more suitable habitat for them. That's what I think. Right. Yeah. Sorry, you have a question there.
I was a facetious question. It was beautifully architected. Surely the architect could design a reef that would snag fish hooks and discourage people.
That's the common complaint with some other fisheries. People go to fish, we must be off. And we're like, oh, that sucks. Yeah.
I was just wondering, again, with the global snapper, when you talked about how you're going to release or start to release, what's guiding where you're going to do the release?
Good question. So we don't know a lot about gold snapper in PNT, especially at least at this big, which is the size that we release them. So what's guiding our release strategy at first is research that's come out of Southeast Asia on gold snapper, which has found that they basically recruit onto kind of coastal rocky reef and then migrate up into estuaries, into the upper mangroves, and they get a bit bigger, and then presumably they make a migration offshore because that's where the big fish. That's where we think they spawn. So I've gone through to Darwin Harbour and found all that subtidal, some intertidal rocky reef, and we're basically plotted that by area. We have a stocking model that we use for barramundi to determine how many fish we put in a unit of area, and we can use that to start. There is a program called Enhanced Fish, which was developed by Kyler and I think Taylor in New South Wales, which you can use to actually really plot how much fish you should put in a certain area, but that requires a lot of ecological data that you don't have. So hopefully with the great program that's coming through, we can update ecological data, we can further improve our stocking ability. We can also have some telemetry to get short-term release survival, but that's still up in the air. And I'd say that the other approach is to stock conservatively. So spread the thing. Don't just go to one bombing and just be like, here's 10,000 fish. There's a lot of effort goes into growing these fish in hatcheries because they're not like barra, which would be dime a dozen. So it takes us a few days to stock fish and spread them out over their habitat, which if it is coastal rocky reef, there's a lot of it. We'll take that. And the Darwin Aquaculture Centre is right next to Channel Island Boat Ramp, so it's really easy logistically to release fish.
Do you have again genetical problems with the hatcheries because I assume they all have this gene?
So, good question. So, same with our barramundi stocking program, we only breed wild fish. We don't have a breeding program. We don't breed F1s. We also, hopefully, we'll start. We do it with our barramundi and hopefully do it with our gold snapper. We'll turn over the brood stock and introduce new genetics. We're going to collect baseline wild genomic data so that we can actually look to see if the stocking is having an effect on the genetic, integrity of the Darwin stock. That's one of those things where you go, look, we don't know if it's going to have an effect until we actually start putting fish in the water. We don't know how many fish we need to put in to have an effect. We can also use genetics in the hatchery to make sure that when we're crossing our females and males, because we have the ability to choose, we can, if we've got one male that's just constantly contributing, we can be like, he's had a few cracks, don't spawn him anymore, start crossing new girls, new boys, to try to improve the amount of genetic diversity that's the program. But it's something that we're definitely we're cognizant of.
Yeah, I guess I'm familiar with sort of threatened freshwater fish stocking stock release strategy. When you've got huge predation and hypervariable habitats to release these juveniles, what sort of training or strategies do you have to help them?
So much. I wish I could do all of this. There's a lot we can do. There's hatchery training, introducing habitat, introducing feed. One thing that we're going to do at DAC, because it's easy, is transition them off of pellets, which is what the fish get now, into prawns, which is from other research, most likely the bulk of their diet involves just prawns. So even just then getting the chemical cues of being like, that's a prawn, I eat prawns now. That kind of stuff is easy for us to do. Honestly, I think, there's a lot of stuff we can do with hatchery training
It just comes down to time, how much time we have to do it. The other aspect is size release. So we're trying to grow our fish out to, the goal is 13 centimetres, which is 20% of their L infinity, and theoretically that is the size at which density independent mortality stops, theoretically. So we're trying to grow our fish to that size. I think the biggest thing for that is hatchery constraints, size, basically production constraints. But we're basically just trying to make it as big as possible to a limit to try to increase that post-release a lot. But both the DNA and tagging and telemetry will hopefully inform those kind of post-release slides so we can get that information and be able to answer those questions.
Yeah, just wondering with the golden snapper to protect the spawning stock biomass, if you have considered any maximum size limits on recreational catch. Is that on the cards or in the song?
I think Barrel disagrees with yeah, but no size limit because of Barrel. It's if you put a limit of 80 centimetres and limit 81 centimetres, I'll be out of 20 minutes water, that fish is dead. Like
yeah. Even at shallow depth, though, I suppose it could save.
I think it's just really hard when doing consistent messaging. We go really hard with messaging of if you're targeting red fish, like you better harvest them. Just the reality, everyone's doing, best practice and fish and handling the well.
Yeah.
So that's feasible, but the reality is that can do that.
The research in what Danilo highlighted as movement, fish movements, it can increase that understanding. If we can identify nursery areas or spawning areas, could be some other measures we can put in place to protect those habitats. So, but we don't have that science at the moment.
Yeah.
Both of the big ones
Very nice. Yes. I was say we've got a few people from fisheries in the room if you guys want to wave.
Yeah, just
before they
just wanted to raise your hand. Justin is our graduate.
Oh yes, current graduate.
So he's doing a lot of work for us at the moment.
Ask about otolith shape.
And soon we'll be looking for a job.
Thank you.
Cool. So a few little announcements before people run off. We have morning tea in about 15 minutes up in Yellow 2. So everyone's welcome to join us there. It's also open day this weekend, so tomorrow. And Real will be doing their thing again in Yellow 2. So if you want to check that out, you're more than welcome. Our next seminar in 2 weeks' time is by BirdLife, and that one is an online only one. So don't show up here, we won't be here, but we will see you online. And then lastly, we have which will be relevant for everyone here, is a marine conservation symposium.
So in a couple of weeks' time, on the 27th, 28th of August, we've got a two full days symposium. It'll be catered. So we do need people to register so we get numbers. First day is aquaculture. And the second day is fisheries, marine ecology and technology. There will be speakers from amongst the NT researchers. Fisheries, just one overlap will be Alex, but he'll be focused in different areas. So do come back and see him again. There'll be NT fisheries, AIMS, CSIRO, and CDU speakers. But the purpose of this is actually an exchange with a couple of Chinese universities. We'll have, so the speaker numbers are split between, it's about 10 from each, 10 from NT and 10 from Hainan and Shantou universities and one Tsinghua University in Beijing. The invite will go out on the real student, staff and adjunct emails, and it will go out. I think everyone, like there's people within fisheries and within AIMS who will receive the invite, and distributor, totally distributed, most will be on the list too. For anyone who might not be on those lists, we do have a small number of hard copy versions. So the invite has on it a QR code for the program and a QR code for registration, so we can get that. Everyone is welcome. Cheers.
All right. Thanks again to our speakers, and we have a little bit of slide. Check names.